So far, we have studied about the formation of endosperm, even the formation of embryo. Even though the initial steps in the development of embryo are one and the same in monocots and dicots, till then a complete formed embryo of monocots and dicots are entirely different. Let us discuss a structure of dicot embryo and its explanation, and even a structure of monocot embryo and its explanation. First, we will discuss about the structure of dicot embryo. Discuss about a structure of dicot embryo. This dicot embryo is an embryonal axis. This is embryonal axis. As it is dicot, it is having two cotyledons where food is reserved. Two cotyledons. As the seed of dicot plants is having two cotyledons, hence the name dicotyledons. This is embryonal axis and two cotyledons. In this embryonal axis, a portion of embryonal axis above the two cotyledons is named as epicotyle. This is said to be epicotyle. This epicotyle terminates into plumule. From that plumule, shoot system is developed. A portion of embryonal axis above the dicotyledons is named as epicotyle. That epicotyle terminates with plumule, and from plumule shoot system is developed. And in the similar way, a portion of embryonal axis below the cotyledons is named as radicle. And this radicle gives rise to root system. The lower portion of embryonal axis beneath the position of cotyledons is named as radicle, and this radicle gives rise to root system, and it is covered by root cap. Once the root system is developed, it has to penetrate through the soil, but at the tip there would be a meristematic, very delicate tissue. To protect that delicate tissue, it should have a root cap. It should have a root cap as well. This is about the structure. With neat label diagram, describe the structure of dicot embryo. Dicot embryo is having embryonal axis with two cotyledons. A part of embryonal axis above the cotyledons is named as epicotyle. That epicotyle terminates with the plumule, and that plumule gives rise to shoot system. In the similar way, a portion of embryonal axis beneath the cotyledons is named as radicle. That radicle gives rise to root system, and it is covered by root cap. As we know, root has to penetrate into the soil. It must be protected. At the tip region only meristematic tissue is there. To protect that, there is a root cap. This is the structure of monocot embryo. As far as structure is concerned, it is little bit complicated as compared to dicot embryo. As it is a monocot embryo, it is having a single cotyledon, and that single cotyledon is named as scutellum. In graminate members, that single cotyledon is said to be scutellum, and this is embryonal axis. Totally, this is embryonal axis. At the lateral side of embryonal axis, there is a single cotyledon. That cotyledon is named as scutellum. That is said to be scutellum. And now, in this embryonal axis, a portion of embryonal axis above the scutellum is named as epicotyle. A portion of embryonal axis which is above the scutellum, above the attachment of that scutellum, is said to be epicotyle. In this epicotyle, there is a plumule with a stem apex. This stem apex is covered by few leaf primordia. Few leaf primordia means very young leaves, leaf primordia. That stem apex or shoot apex is covered by few leaf primordia, and in turn is covered by a hollow foliar sac that is named as coleoptile. Coleoptile. A portion of embryonal axis just above the position of that scutellum is named as epicotyle. No doubt it terminates into plumule, where we do get, instead of saying stem apex, even you can say shoot apex. Shoot apex. It is covered by few leaf primordia. Leaf primordia, and in turn it is covered by a hollow foliar structure named coleoptile. That is coleoptile. Here shoot apex is not naked. It is covered by a leaf primordia as well as coleoptile as well. And a portion of embryonal axis below the position of scutellum is named as radicle. Radicle. And that radicle gives rise to root system. and this radicle is covered by a root cap. Covered by a root cap. And a radicle and a root cap are in turn covered by a undifferentiated sheath named coleorhiza. Coleorhiza. That radicle and root cap are covered by undifferentiated sheath, a covering named coleorhiza. This is about the structure of monocot embryo. Monocot embryo, its structure is a little bit complicated as compared to dicot embryo. As it is, monocot embryo is having single cotyledon. That single cotyledon in the graminae members is said to be scutellum. Scutellum. And this scutellum is present at the lateral side of embryonal axis. Lateral side of embryonal axis. In this embryonal axis, a part of embryonal axis above the position of scutellum is named as epicotyle. That terminates into plumule. And the portion of embryonal axis below the position of scutellum is named as hypocotyle. Lower portion. And here we do get a radicle that gives rise to root system and is provided with the root cap. And one more additional sheath around this radicle and root cap, that is a undifferentiated sheath named coleorhiza. Coleorhiza. And this epicotyle, a part of a embryonal axis above the scutellum is epicotyle, which gives rise to plumule. Here we could see a shoot apex. Shoot apex that is formed from plumule only. And this shoot apex is covered by few leaf primordia, primitive leaves. Is covered by few leaf primordia and is in turn covered by a hollow foliar structure named coleoptile. Coleoptile. Here, both shoot apex and root apex are covered by extra coverings or sheath. That root apex is covered by coleorhiza, whereas that shoot apex is covered by coleoptile. Yes. Now, it's time to discuss about seed. I have taken here the structure of monocot seed. Seed, is nothing but a fertilized ovule. After fertilization, ovule becomes seed. After fertilization, ovule becomes seed. And remember, seed is a final product of sexual reproduction. It's a final product of sexual reproduction. As ovule becomes seed after fertilization, the integuments, the coverings of ovules become seed coat. They become seed coat. Seed, we do have two types of seeds. Albuminous seeds and non-albuminous seeds. Albuminous seeds and non-albuminous seeds. Albuminous seeds are the seeds in which the endosperm is persistent or retained. Already you know about endosperm is a nourishing tissue, is a triploid tissue. It provides food to the growing embryo. But in some seeds, the endosperm is remained persistently. And such seeds which are with endosperm are named as albuminous seeds. The example for albuminous seeds are all the cereals like maize, jawara, wheat, rice and coconut, sunflower, even castor. They are all albuminous seeds in which the endosperm is there. Already we know about in coconut both free nuclear endosperm, that is in the form of coconut water, and there is a cellular endosperm which is there in the form of kernel, a white kernel. Both of them are there in that coconut. Even its work is to provide food to the growing embryo till it is remained in the seed. Such seeds are named as albuminous seeds. And the non-albuminous seeds are the seeds in which endosperm is absent. It means that endosperm is used up at the development of embryo. Example for non-albuminous seeds are papillonosis members like pea, beans, groundnut, where we don't find any endosperm at all. Those are non-albuminous seeds. And one more interesting thing, even a little of nucellus. What is nucellus? That is the internal structure of ovule, in which one of the nucellus cell becomes the embryo sac. Even in some seeds, nucellus is present or persistent, and such seeds are named as perisperm. Perisperm. This you could see in case of pepper and beet, where the presence of nucellus in the seed, not only endosperm, even the presence of nucellus. Actually, once the embryo sac is completely formed, nucellus gets disappeared at the time of embryonic development only. But here, the presence of that nucellus, that is said to be perisperm as well. An example for that is pepper and beet. Pepper and beet. Let us see. Already we know seeds are the final products of sexual reproduction, and seed is formed from the ovule after fertilization, and coverings of ovules named integuments, they are becoming seed coat, a hard, tough seed coat. And the structure which I have drawn is a monocot seed from cereals, and this seed is a single seeded fruit also, because the seed is there in a fruit, a single seed in single fruit, so that this outermost covering, it is named as pericarp. Pericarp. Pericarp is a covering of fruit. Is a covering of fruit. But this is a single seeded fruit. That's why even that fruit covering is also mentioned. Next to that is a hard and tough seed coat. In this fruit, pericarp and a seed coat, they are inseparable. They are attached to each other. But in case of sunflower, that is also a single seeded fruit, but the pericarp, a fruit coat as well as a seed coat, they get separated. They get separated. You might have seen a sunflower seed. We used to say seed. Actually, that is a single seeded fruit. A black covering, if you just opens it, you will get a single oil seed, where that pericarp and seed coat get separated. But in case of cereals, that pericarp, a fruit covering and a seed coat, they are inseparable. And this is endosperm. As I have mentioned, cereals, they are albuminous seeds. They have retained endosperm. They have retained endosperm. And then this part is a cotyledon, scutellum. There is a single cotyledon in monocot seeds, and that single cotyledon is named as scutellum. And this scutellum, just at the lateral side, embryonal axis is there, and that embryonal axis is having primule, shoot apex, covered by leaf primordia and a hollow foliage structure, coleoptile. Because this is embryo, already we have seen the structure of embryo, and this is radicle, gives rise to root system, covered by root cap, in turn covered by an undifferentiated sheath named coleorhiza. And there is a minute pore, a very small pore is here. This is nothing but a micropyle of ovule. After fertilization, ovule becomes a seed, integuments become a seed coat, and a micropyle becomes a small pore. Through this small pore, oxygen and water enters at the time of germination. When this seed starts germinating, through this water and oxygen enter and the seed gets germinated. It's about the structure of seed. The example is monocot seed. And among the seeds, based on presence or absence of endosperm, we have made albuminous seeds where endosperm is retained, and non-albuminous seed where endosperm is not at all present. With the example, even some exceptional cases, seeds from pepper as well as beet, where nucellus is also present in the seed. That is said to be perisperm. And the structure in quick glance. I have drawn the structure of cereals, and where cereal seed, where they are all one-seeded fruits, so that the outermost covering is pericarp. Next to that is seed coat. It's hard and tough. But in cereals, pericarp and seed coat are inseparable. They cannot be separated. And as it is a luminous seed, we do see the endosperm. And as it is a monocot, we do get a single cotyledon. That single cotyledon is said to be scutellum. At its lateral side, there is an embryonal axis, and upper portion of embryonal axis is having primum. That is a shoot apex covered by leaf primordia and a hollow foliar structure named colluteine. And lower portion of embryonal axis is a radicle which gives rise to root and is covered by the root cap, and which is in turn covered by a undifferentiated sheath named coleorhiza. And once the seeds are formed, they get dried. Even their metabolic activity is also decreased. They lose the water. Remember, these dry seeds, they do have 10 to 15% of moisture as far as the total weight is concerned. And they will enter into dormancy period, resting period. When they enter into dormancy period, seeds never get germinated during their resting period. And the duration of the dormancy period varies from seeds to seeds. They may take the dormancy period of 1 month, even a year. Even there are some record of old seeds we do have. Dormancy period means resting period. As soon as they are formed, those mature seeds lose water and become dry. Just 10 to 15% of moisture will be remained in them, and they enter into resting period. That is said to be dormancy period as well. That is said to be dormancy period. And once they enter into dormancy period, their next germination starts when they get the favorable conditions like moist oxygen and suitable temperature. They get the water and oxygen through the small pore and they start germinating. And there are 2 examples. One is lupine. Its botanical name is Lupinus arcticus. Lupinus arcticus. Its seeds were obtained during excavation in Arctic tundra. During excavation means utkanana. The seeds of Lupinus arcticus are obtained in Arctic tundra. Those seeds, after 10,000 years, started germinating, so that their dormancy period is 10,000 years. They were submerged for about 10,000 years. At the time of excavation, seeds of lupin were obtained. And those seeds, after 10 years of dormancy period, started germinating. And one more example is there, that is date palm. Its botanical name is Phoenix dactylifera. Phoenix dactylifera. And the seeds of this date palm were obtained during archaeological excavation of King Herod's palace near Dead Sea. There was an archaeological excavation, Utkhana, of King Herod's palace, where nearby Dead Sea, where seeds of date palm were obtained. The surprise thing is, those seeds started germinating after 2,000 years. So that the dormancy period for them is 2,000 years, date palm. And the dormancy period for lupin is 10,000 years. And as far as number of seeds is concerned, in cereals, just one seed in a fruit. And there are many examples like orchids, innumerable number of seeds are there, very minute seeds. Even ficus, banyan tree, we do get, if you collect all the fruits of one banyan tree, there are many billion seeds are there in all the fruits as well. Even some parasitic angiosperms like Orobenche and Stygra. There are also innumerable number of seeds. The number starts from 1 to thousands, millions are there in one fruit as well. It's about seeds. We have studied about fruits in first year. As we are dealing with the reproduction in flowering plants, it is time to know a brief account of fruits also, in which the seeds are there. Already you know that after fertilization, ovule becomes seed, integuments become seed coat, micropyle becomes a pore in which oxygen and water can enter into the seed at the time of germination. In the similar way, after fertilization, ori becomes fruit. Ori becomes fruit. Let us have a brief account about fruits. What are fruits? Fruits are matured, ripened ori. Matured, ripened and fertilized ori. It means after fertilization, a ori becomes fruit. Ori becomes fruit. It's a general definition for the fruit. Fruit is a matured, ripened, fertilized ori. Fertilized ori. And fruit coat is named as pericarp. That fruit coat is named as pericarp. And this pericarp may be undifferentiated or is differentiated into epicarp, mesocarp and endocarp. Even it may be undifferentiated. No distinction. Pericarp is a fruit coat. It may be undifferentiated or is differentiated into outer epicarp, middle mesocarp and inner endocarp. And if the pericarp is undifferentiated, that is, the fruit is said to be berry. Where pericarp is undifferentiated. If it is differentiated into epicarp, mesocarp and endocarp, then that fruit is said to be drupe. An example for berry, tomato. An example for drupe, coconut and mango. Coconut and mango. In tomato, total pericarp is edible and is undifferentiated. And mango, is a drupe where you can notice, if you see ripened mango, outer epicarp, yellowish green. When it is raw, green and gradually becomes yellowish, and that is epicarp. And fibrous, juicy mesocarp, and inner endocarp, very hard endocarp. Within that endocarp, we can see a single seed as well. A seed is there inside. And in case of coconut, and in drupe, that is mango, which part is edible? That mesocarp, which is fibrous and juicy. And in case of coconut, all these three parts are there, but these three parts are not edible. A yellowish green thin covering on coconut fruit, that is epicarp, and a dry fibrous mesocarp, and a hard part inside is an endocarp. And which part is edible in coconut? Endosperm. Both liquid and solid endosperm. Both free nuclear and cellular endosperm. In mango, mesocarp is edible, and in coconut, these three things are not at all edible. The edible part is endosperm. It's about how this pericarp is differentiated and undifferentiated. In almost all the fruits, we do see ori becomes fruit. Ori becomes fruit. Any fruit which is formed from ori after fertilization is a true fruit. Is a true fruit. What is true fruit? Is a fruit which is formed strictly from ori. Other floral parts will shed off. They won't remain in the fruits. Only ori becomes fruit after fertilization. Such fruits are named as true fruits. Example for this: mango, coconut, all these things. Most of the fruits are true fruits. If the fruit is formed from any floral part other than ori, if fruit is formed from any floral part other than ori, such fruits are named as false fruits. False fruits. What is false fruit? A fruit which is formed from any floral part other than ori. Such fruits are named as false fruits. The best example is apple. In apple, thalamus, a floral axis, thalamus becomes fruit. You might have seen in apple, at the center there is a ori with seed. That never becomes the fruit. The whole thalamus becomes fruit. It's a false fruit. But there are some fruits which are formed without fertilization, where ori becomes a fruit without fertilization. And such fruits are named as parthenocarpic fruits. Best example, banana and a grape, where no seeds at all, because no fertilization at all. Ori becomes a fruit without fertilization. Such fruits are named as parthenocarpic fruits. An example, banana and a grape. Banana and a grape. With this, we have come to the final conclusion. Let us discuss about two terminologies. One is apomixis and polyembryonine. Apomixis and polyembryonine. What is apomixis? It is taking place usually in Asteraceae and even in some grasses. Asteraceae member is a sunflower, where apomixis takes place. What is apomixis? Is one where the formation of seeds without fertilization. No fertilization. Without fertilization, ovule becomes a seed. And that is named as apomixis. A typical 1 mark question. They would ask, what is apomixis? A formation of seed from the ovule without fertilization. Formation of fruit without fertilization of ori is parthenocarpic fruits. In the similar way, formation of seed from the ovule without fertilization, that is named as apomixis as well. And example, in case of Asteraceae members like sunflower, even grasses, we can see apomixis. Even in some species, there would be a diploid egg cell in embryo sac, because where there is no reductional division. Egg cell is formed without reductional division, so that it is diploid, so that the egg cell directly becomes the seed without fertilization. Even that is another way of the formation of apomictic seeds. Apomictic seeds. And apomixis, is a type of asexual reproduction. But it looks like sexual reproduction as well. Because here seeds are there. They are formed without fertilization. And next one is polyembryonine. Already we know that in a single ovule there is a single embryo sac with single egg cell. If you say one ovule, in that ovule there is a single embryo sac. In that single embryo sac there is a single egg cell. So that from that one ovule we do have one seed as well after fertilization. In case of some species of mango and citrus, some nucellar cells, nucellus, that is there in ovule, some nucellus cells present side by the embryo sac get enlarged and become embryo sac. Some more nucellar cells side by the embryo sac in a single ovule get enlarged and become embryo sac. So that in such seeds, we do find more than one embryo. In one embryo sac means one egg cell is there, after fertilization one embryo. But here, in case of some species of mango and citrus, some nucellar cells around the embryo sac get enlarged and become embryo sac. In them also, egg cell is there, they too get fertilized. Then we do have more than one embryo in a seed. That is said to be polyembryonine. Poly means many. Many embryos in a seed. Polyembryonine is a condition we do see in some species of mango and citrus, where some nucellar cells around the embryo sac get enlarged and become embryo sacs, so that there is more than one embryos in the seed, and that is said to be polyembryony. Polyembryony. And now a maximum using of hybrid seeds nowadays. But the nature of hybrid seeds is that once we use the hybrid seeds, we will get the yield. But those seeds, if you go for second generation plantation, you won't get that hybrid characters, because hybrid characters will be segregated. In order to avoid that, because every time during plantation, farmers have to purchase the hybrid seeds, because they won't be used generation after generation. But lots of work by plant breeders in apomixis and also polyembryony. Apomixis, that is a type of asexual reproduction where seeds are formed without fertilization, so that if those seeds, this kind of apomictic hybrid seeds are used, there would be no chance of segregation of characters, so that the same seeds can be used for many generation as well.